Hey everyone, so for this video we're going to be doing an experiment, and this experiment is getting the genetic info from a banana. So the question is it possible to see DNA? Is DNA soluble in alcohol? In this experiment I will be trying to figure this out by taking the DNA of a banana and placing it in alcohol, and I'll show you how to do that in a second. Before starting this experiment, it's important to take note of the different properties of DNA. DNA is the genetic code for making proteins, which then build up organisms. It is stored in the nuclei and mitochondria of cells. Also, for this experiment, it's necessary to understand polarity and solubility. Chemical Dictionary defines electronegativity as the measure of how strongly atoms attract bonding electrons to themselves. Polarity is when there are different electronegativities in a molecule and the bonding electrons are not equally shared. When an atom has a high electronegativity, it will likely have a partial negative charge. This affects the attraction between atoms. The best example of this is a water molecule, H2O. The two hydrogen atoms have a partial positive charge, while the oxygen atom has a partial negative charge. This is because it is electronegative. Due to the differences in charges, the hydrogen atom's bonding electrons are attracted to the oxygen atoms, but the sharing of electrons is not equal. The partial charges and unequal sharing of electrons makes water a polar molecule. Polar molecules are attracted to other polar molecules due to the partial negative and partial positive charges and will bond to each other. That being said, polarity affects solubility, which is whether one substance, the solute, can be dissolved by another substance, the solvent. For example, salt is a polar molecule and so is water. When salt is placed in water, the water will dissolve the salt because they are attracted to each other. Oil is nonpolar and therefore will not dissolve in water. In our experiment, however, we want to find out if DNA can be dissolved in alcohol. Is alcohol and DNA attracted to each other? Let's find out. For this experiment, you'll need a peeled ripe banana, a half cup of hot water, a teaspoon of salt, a resealable freezer Ziploc bag, a half a teaspoon of dishwashing soap, rubbing alcohol, a coffee filter, a glass cup, and a small stirrer. I just used a spoon. So first, you're going to want to place the bottle of rubbing alcohol into the refrigerator because it won't be needed until step 9. Next, cut off a portion of the peeled banana. I cut off like an 1.5-inch portion of the banana from the end. Once it's cut, place the banana into the bag. Move the banana piece into the corner of the bag, seal the bag, and then mash the piece up smoothly. Make sure there are no chunks left. For step 4, set the bag off to the side for a moment. Pour the teaspoon of salt into the hot water. Stir the solution to create salt water. Take the bag once again and open it, and pour the salt water inside. Afterwards, seal the bag again. Proceed to mash up the banana and the salt water until it's fully mixed. Now pour the half teaspoon of dishwashing soap into the bag and reseal. Mix it up fully and try not to create too much foam. Place the bag off to the side once again. Take your cup and coffee filter. Put the coffee filter on the inside of the cup and create a filter for the liquid. The liquid is going to go through the filter and into the cup. It's recommended that the coffee filter edges are folded over the cup's rim. Open the bag and gradually pour the bag's contents onto the filter. Do not do it all at once, otherwise the cup will overflow because it cannot filter fast enough. Wait for the contents to drain fully. This may take about 8 to 10 minutes. After all the liquid is filtered through, throw out the contents left on the filter as they are no longer needed. You can now take the rubbing alcohol out of the fridge. Gently pour the alcohol into the cup with the liquid. Create a layer of alcohol about 1 inch or 2. Let the cup sit for about 8 minutes. A substance will start to bubble and clump up. This white substance is the DNA of the banana. Lastly, place the stirrer into the cup. Try to take out the DNA from the cup if you want, but you can just keep it in. Then, drink it. 'Cause why not? This is a safe experiment for kids. Now, you might be wondering, what just happened? Essentially, it went like this. First, the DNA is wrapped around a histone, which is a protein. The salt we added to the banana water mix got rid of these histones, and also made the DNA less soluble. Next, the dishwashing soap. Cells in their nuclei are surrounded by a membrane barrier. These membranes are composed of fat molecules. The soap will break down these membranes, releasing the DNA from the cells. After all that is done, we added the alcohol. Since all the DNA has been exposed, the DNA is floating in the alcohol. It will go to the top of the alcohol because it is not soluble in alcohol. That being said, our question has been answered. Also, if you add too little of these things, the experiment will not work well. It's important to measure things properly, like I tried my best to do, in order to get the expected results. DNA is used for many things, especially in the forensic science field. Although bananas may not be committing crimes, experiments like these have led us to other discoveries with our own DNA. I hope you guys learned something today and had fun doing this experiment with me. Thanks guys, and I'll see you all in the next video.